Amino acids are far more than the simple structural building blocks of muscle tissue. In the realm of biological longevity, cellular repair, and metabolic optimization, these fundamental molecules serve as central signaling regulators governing macromolecular synthesis, adenosine triphosphate, ADP, production within the mitochondria, cellular signal transduction, and the direct epigenetic regulation of human gene expression. Every single cell in the human body relies on a precise, highly balanced pool of intracellular amino acids to maintain everyday homeostasis and execute vital cellular repair processes. When the dietary supply of protein fails to deliver the optimal ratios or quantities of specific indispensable amino acids, a hidden, systemic metabolic insufficiency occurs. Emerging clinical and preclinical evidence suggests that targeted amino acid supplementation can correct these specific metabolic deficits, optimizing physiological function and promoting a longer, healthier lifespan. However, navigating this complex cellular landscape requires strict precision. However, robust clinical research demonstrates that these regular consumption patterns frequently fail to meet the elevated metabolic demands brought on by aging, intense physical activity, chronic metabolic stress, or illness. As the human body undergoes the biological aging process, a frustrating phenomenon known as anabolic resistance sets in. This condition means that older muscle and organ tissues require a significantly higher concentration of essential amino acids, particularly the branched-chain amino acid leucine, to trigger the exact same level of protein synthesis that a younger organism achieves with ease. When the diet lacks these elevated levels, the body progressively downregulates its maintenance pathways, accelerating muscle wasting, compromising immune function, and reducing mitochondrial efficiency. In a healthy system, this delicate balance between protein synthesis and autophagic clearance preserves youthful cellular function. However, a modern, disorganized diet can disrupt this rhythm. By continuously flooding or starving the system without regard for metabolic timing, we trigger cellular confusion, driving either accelerated aging through overactivation or tissue degradation through chronic nutrient deprivation. Beyond general protein synthesis, individual amino acids perform highly specific, non-proteogenic roles that directly impact cardiovascular health, immune defense, and neurological function. For instance, the amino acid L-arginine serves as the direct precursor for the production of nitric oxide, a powerful signaling molecule that commands the smooth muscle cells of our blood vessels to relax. This relaxation, known as vasodilation, is completely necessary to maintain healthy blood pressure, ensure optimal oxygen delivery to distant tissues, and prevent endothelial dysfunction. Similarly, amino acids like L-glutamine act as primary fuel sources for rapidly dividing cells, particularly the enterocytes lining our digestive tract and the white blood cells of our immune system. During times of metabolic stress, the body's internal demand for glutamine can easily outpace its capacity for synthesis, leading to a compromised intestinal barrier and an impaired immune response. By strategically utilizing precise, bioavailable amino acid supplements, we can directly target these individual biological pathways, correcting localized tissue deficits and optimizing systemic resilience against environmental stressors. Furthermore, recent scientific breakthroughs have revealed that amino acids exert a profound, direct influence on the human epigenome, altering gene expression without modifying the underlying DNA sequence. Specific amino acids, such as methionine and glycine, are core components of the 1-carbon metabolism pathway, which generates S-adenosylmethionine, the universal methyl donor for DNA and histone methylation. This biochemical process acts as a master volume knob for our genome, silencing pro-inflammatory genes and activating vital longevity and DNA repair pathways. If the cellular pool of these specific amino acids becomes depleted or severely imbalanced, the fidelity of our epigenetic landscape degrades, leading to aberrant gene expression patterns that mirror the classic signatures of accelerated cellular aging. This deep intersection between nutrition and genetics emphasizes that we cannot treat amino acids simply as passive fuel. They are active molecular signals that constantly rewrite the operational instructions of our cells. However, because amino acids are highly potent biological agents, more is not always better. The clinical literature warns extensively against the dangers of unregulated, excessive supplementation that overshoots the body's natural processing capacity. When an individual consumes random, massive doses of isolated amino acids, they risk overwhelming the specialized transporters in the gut and the blood-brain barrier, effectively blocking the absorption of other equally vital nutrients. This competitive inhibition can create secondary, localized deficiencies inside the brain and nervous system, disrupting neurotransmitter synthesis and causing cognitive fatigue or mood imbalances. Additionally, an overload of amino acids forces the liver and kidneys to work under high pressure to clear the excess nitrogen through the urea cycle. If this pathway is continuously strained, it can lead to a toxic buildup of ammonia in the bloodstream, inducing oxidative stress, damaging delicate endothelial tissue, and placing a dangerous metabolic burden on the entire organism. To prevent these toxic overloads, modern nutritional science relies heavily on establishing rigorous safety parameters derived from carefully controlled preclinical and clinical trials. The primary metric used by researchers is the no observed adverse effect level, which determines the absolute maximum dose of a specific substance that can be consumed without producing any measurable biological harm. From this baseline, scientists calculate the upper level of safe intake, providing a clear, mathematically sound boundary for therapeutic supplementation. Clinical data demonstrates that while the human body can tolerate remarkably high variations in whole protein consumption, isolated single amino acids possess highly specific, rigid safety profiles. For example, while certain amino acids like leucine have a relatively wide safety margin, others can trigger metabolic distress at much lower doses. Respecting these boundaries is a non-negotiable rule of advanced longevity medicine. It ensures that we maximize the targeted therapeutic benefits of nutrient signaling while avoiding the dark side of metabolic toxicity. Furthermore, we must look closely at how specific combinations of amino acids can either maximize or interfere with human brain chemistry. The blood-brain barrier utilizes specific gatekeepers known as large neutral amino acid transporters to let nutrients pass from the blood into the brain. If a supplement formula is completely saturated with just one type of amino acid, it effectively crowds out all others at the gate. For instance, if you take too much of a single branch-chain amino acid, it can block tryptophan and tyrosine from entering the brain. Because tryptophan is the mandatory precursor for serotonin, and tyrosine is the precursor for dopamine, this simple competitive inhibition can cause a sudden crash in mood, focus, and mental drive. This is why multi-targeted longevity formulations must be mathematically balanced. We must ensure that optimizing muscle or metabolic health does not inadvertently cause a neurochemical deficit that triggers brain fog, depression, or sleep disturbances. In addition to brain health, advanced clinical research is now focusing heavily on how amino acids preserve and regenerate the extracellular matrix, which holds our tissues together. The structural integrity of our skin, blood vessels, joints, and organs relies entirely on the continuous synthesis of collagen and elastin. This regenerative process requires a highly specific and abundant supply of conditional amino acids, most notably proline, hydroxyproline, and glycine. Glycine, in particular, acts as a major bottleneck for collagen production. While the body can synthesize some glycine internally, aging and chronic inflammation vastly deplete these stores, leading to the progressive thinning of blood vessels, joint degradation, and the visible signs of skin aging. By providing the body with targeted, safe doses of these specific structural amino acids, we provide the raw materials required to rebuild the structural scaffolding of the body, directly protecting vascular elasticity and joint longevity from the inside out. Looking to the future, the definitive conclusion of this extensive research points toward a highly sophisticated, individualized paradigm for human health and clinical nutrition. We are rapidly moving away from generic, one-size-fits-all daily protein recommendations, transitioning instead into an era of hyper-personalized amino acid modeling. Future medical protocols will likely utilize advanced metabolomic screening to map an individual's unique plasma amino acid profile in real time. By identifying specific, subtle deficiencies caused by genetic variations, lifestyle factors, or age-related metabolic shifts, practitioners will be able to design custom-tailored amino acid formulas. These precise interventions will serve to seamlessly repair broken metabolic pathways, preserve structural lean mass, quiet systemic neuroinflammation, and maintain the operational integrity of the epigenome. Ultimately, by mastering the precise balance, timing, and safety thresholds of these vital molecular messengers, we can transform basic nutrition into a predictable, mathematically precise tool for life extension, physical resilience, and long-term cognitive preservation.